well, we only had 2 quizzes on chapter 3, right? The first quiz was on carbohydrates and lipids, and the second quiz, which was on proteins and functional groups, we went over yesterday. So if, I'm gonna pass back the first quiz, if there's something there that you want to review, I'm gonna give you a few minutes to do that. But we did go over these together already.
Did you have an actual one that has
These are the questions that went with that. Does everyone have enough time to look at these? Would you pass the question sheet up? Now, what should you be reviewing from chapter 2? Not that there will be very many questions. most of the tests, as I said on Tuesday, will be on chapter 3. But chapter 2 is the elementary chemistry, right? What's the important thing in that chapter? It's understanding
Chemical
different
bond.
kinds of bonding, right? So ionic bonds versus covalent bonds, either polar or non-polar. And we looked at that probably in greater depth than you saw it last year with Mr. Donnelly. Particularly, we talked about electronegativity values and the use that you can make of the electronegativity value. I don't know what that is, but get rid of it. The electronegativity values can be used to determine the kind of bond between any two what? Any two atoms, right? As long as you've memorized 0.5 and 1.7, then you take the difference in electronegativity values. Also in chapter 2, we reviewed the difference between an isotope. What are isotopes? Yes.
Two of the same element, or like two atoms of the same element that have different numbers of neutrons.
Right, they have different numbers of neutrons, and as a result, they have different masses, correct? And how do you get the mass number of an atom? By adding the protons and the neutrons, but not the electrons, right? You remember we looked at where you would put the mass number and the atomic number. So if I was talking about carbon, for example, well, carbon has atomic number 6, and a common mass, a common isotope is carbon 12. But there's also carbon what? 14. Right? So how many neutrons does carbon 14 have? 8. How many neutrons does carbon 12 have?
6.
6. How many neutrons does carbon 13 have?
7.
7. But they're all carbons because they all have the same what number?
Atomic
Same
number.
atomic number, the mass number difference, right? Those are isotopes. Not to confuse that with what we've been talking about in this chapter 13 with isomers. Isotopes. They sound the same, right? Same beginning letter and I. What are isomers? Yes.
Molecules that have the same molecular formula but are arranged in different ways.
Correct, and so they are different compounds. And of course, we see a lot of that in organic chemistry. For example, many sugars, right, have what? Isomers, like fructose and glucose and galactose. They're all C6H12O6. They're hexose sugars, but they're not the same sugar. They're only isomers of each other. How about the 3 disaccharides, maltose, sucrose, and lactose? They all have what formula?
Glucose.
C12H20OE11 because of the lack of water there, yes? They're also isomers of each other, right? And your familiarity with the periodic table, everybody knows the difference between an acid and a base. We reviewed that in chapter 2, right? Acids donate hydrogen ions, whereas bases can either what or what? They can accept hydrogen ions like the amino group does, which is why it acts like a base, right? Or you can donate what?
Hydrogen.
You can donate hydroxide ions, right? Either way, the substance is a base, right? All right, any questions before we move on and talk a little bit more about transcription and translation? Now, what is the difference between transcription and translation? We talked about this yesterday. What are you doing in transcription? What goes on in transcription? Jack?
The RNA is made to match the DNA strand.
So you're converting what to what then?
DNA to RNA.
You're converting the nucleotide sequences of DNA to the nucleotide sequences of RNA. Does anybody need a transcribing dictionary? No, it's in your head, right? You know that adenine pairs with what? You know, with uracil, that thymine pairs with adenine, cytosine with guanine, and guanine with cytosine. Now, that dictionary is in your head. You know how they pair up. So that if I wanted to convert the DNA to the RNA, I would know that by heart, yes? So, the second step is called translation. Now what's going on in translation? Now I'm going from nucleotide sequences of RNA to what? amino acid sequences of a polypeptide chain. You see how they're not the same process? Don't confuse the word transcription with translation. They cannot be interchanged. They don't mean the same thing, right? Always, when I'm going from DNA to RNA, and notice that the RNA here is called messenger. See the M in front of it? It's called messenger RNA, right? Because it's going to convey the message to the who out in the cytoplasm that's going to be making the protein. What is the organelle that does that? It's the ribosome, right? That's why it's conveying the message, because DNA is going to be inside the what? It's in the nucleus. Whereas the ribosomes are outside the nucleus. Right? So in any event, what does this first arrow represent? Trans what here? This is transcription. I'm transcribing from DNA to RNA. But when I go from RNA to an amino acid sequence, polypeptide chain, that is called trans what? Translation. Here's a better diagram. This thing represents the cell. The yellow thing is the cell. This big purple blob, that's the what? That's the nucleus, which of course is where we house all of our what? All these chromosomes, all this DNA is housed safely in the what? It's in the nucleus. And does that DNA ever leave the nucleus? Absolutely not, because how many copies do we have of this genetic information?
A lot.
You just get one copy of the DNA, that's it. You get one copy of the DNA. That's why we call this the master what? It's the master genetic code. It's the master genetic code. Are we ever going to let that DNA out of the nucleus? No way, right? That DNA stays in the safety of the nucleus. But where are the ribosomes? Ribosomes aren't in the nucleus, they're out here in the what?
Membrane.
They're in the cytoplasm. Right? So if the ribosome is going to be able to take the messenger RNA and turn it into a polypeptide chain, somehow the information has to be conveyed from the nucleus to the what? To the cytoplasm, and that's the function of what molecule? That's what the RNA is doing. You see, the RNA is nothing but a photocopy, right? Isn't it a copy? But is it a copy of the entire DNA? Is messenger RNA a copy of the entire DNA? No. What is it a copy of? What is it a copy of?
The part of
A nucleotide.
It's the copy of the portion of the DNA that tells me how to make one what? One polypeptide chain. You get it? I'm not going to copy the whole thing. All I want to do here is, let's say I want to make insulin, right? This protein insulin. Well, this DNA, the only thing the DNA contains is the information I'm making insulin? There's 25,000 genes here, yes? We only want to copy one gene, the gene that says this is how to make what? Insulin. So do you see how the messenger RNA is a copy of one tiny, tiny, tiny little piece of the DNA? Do you get it? It's one little piece. And that gets sent out to the cytoplasm. Now, what if that messenger RNA gets damaged? Do we really care? No, because could I make another copy? As long as I have the master genetic code safely inside the nucleus of the cell, can I make as many copies as I want? Sure I can. That's why, what do we allow to leave the nucleus is the what? The messenger RNA goes out there. If the messenger RNA gets damaged or gets messed up, who cares? I can always make a what? Another copy. You get it? You see why we don't ever let the DNA go out there? The DNA is the master copy. Here's the analogy that I use that I think might make sense to you. You know, let's say that your family comes into a lot of money and you decide, you know what, we're going to build the best lakeside home. You know, we bought the best lakeside property on Lake Massabesic or Lake Winnipesaukee, and we're going to build this incredible house, right? So the first thing you do is you go to an architect, right? And the architect designs, right, all of the plans for this beautiful home, right? And when the architect is done, the architect gives you the master what? The master blueprints, right? How many copies does the architect give you